Okay, let's make a start then. So for the first of these talks, the idea with these bite-sized bioinformatics things is that we wanted to find a way to keep our training program going whilst we were all sort of spread around the country and sitting in various living rooms. This sort of Zoom technology seemed to work quite nicely to be able to do it, but we didn't want to commit to doing large courses in the first instance. So we thought a series of these sort of short form trainings to pick on a little topic in bioinformatics and spend probably no more, well, certainly not today, no more than an hour going through this, allows us to sort of make a start on this and work out the technology and hopefully give a little bit of insight into a whole variety of topics. To kick off today then, we're going to look at something inside the SeqMonk software. So hopefully you're all sort of basically familiar with SeqMonk. So it's a desktop application for analyzing, well, a variety of different high throughput sequencing data types. So really anything that produces mapped sequence, so positions in a genome, is suitable for putting into this, and you can analyze RNA-seq, ChIP-seq, ATAC-seq, all of those kinds of data inside it. Today we're going to look at one particular aspect of this, which is the use of Vistories. And Vistories are something that have appeared in SeqMonk relatively recently, probably within the last year. They actually came about from a project at the Cambridge area bioinformatics hackathon. And they're an interesting way to sort of combine the flexibility of a graphical program like SeqMonk with more of the sort of rigorous recording and reproducibility that you would get from using something like R. So today all I'm going to do is to talk you through what Vistories are, to show you how they work in practice, to give some sort of tips and stuff for how to use them, and then to finish up by showing you a thing called Vistory database, Vistory DB, where we're actually starting to collect some of these. So not only can we use these for recording analyses, but we're also using them as part of the training as well. I'm going to switch across then to open up a SeqMonk project that I've already prepared for this. So, ooh, it's interesting. Can you see that? Because that looks really weird on here. Let me just stop sharing that. I'm going to try once more because something didn't look right. Aha, okay, that's why it looks weird. Okay, right, let me try that once more because it looks better now. Hey, that's better. Okay, so this is a SeqMonk project. And SeqMonk has a number of advantages, but also a number of disadvantages over other programs for doing sort of larger scale NGS analyses. The good thing about this is that it's a nice sort of flexible system that allows you to look at a large amount of data. You can zip around a genome very easily. You can look at lots of different things. You can do all sorts of plots very quickly. So it allows you to try a whole load of stuff out. But the disadvantage of that is that the flexibility from being able to try all this stuff can sometimes mean that you can spot things or you can figure things out, but you don't have a nice sort of rigorous way to record that or share that with other people. And Vistories are an aim to try and solve that problem. Within SeqMonk itself, there is a number of things that the program will sort of automatically record to try and give you a sort of proper record of what it was that you did. And some of those have existed in the program for quite a long time. So for example, if I go and look at a data set that was imported into here, if I just double click on that, behind the data set itself, there's a whole load of information. So for example, this is the original file that data set came from, so I can track exactly where it was from. It records the options that were selected when the data was imported, and various other sort of metrics around there. Likewise, when we've done quantitations on here, those also have information stacked behind them. So here I've done a standard RNA-seq quantitation. This is an RNA-seq experiment, not that it really matters. But here, behind all probes, it tells me how the quantitation was done. So this is transcript features over mRNA, quantitative with the RNA-seq pipeline, log transformed, and whatever, any other modifications I've made. Likewise with the filters that I've applied, so I've got a couple of statistical results that I've combined in here already. Those two, if I double click on them, have a proper formal description of whatever it was that was done with them. So the program itself is keeping all of this information for me, but in a format where it's not kind of all in one place. I have to go digging for it, but the program is actually recording it. Something else we've had in the program for quite a long time is that although we have these sort of formal sort of descriptions of what, for example, a filter did, underneath there's a little thing that says comments, and you have for a long time been able to click on or right click on any probe list on here and then say edit comments, and you can put then an arbitrary text in here to say anything you like, and it now means that associated with that list will be the comments that you've put on, because we realized quite a long time ago that it's not enough just to describe what it was that you did. Really, the interpretation also requires you to be able to annotate, well, why did I do that? What was I aiming to do? What did I think of the results? So that commentary around your analysis is really important as well. Vistories then were a step up from this. So Vistories are created automatically with any SeqMonk project. So a Vistory, you don't need to do anything special to make it happen. It's just kind of running in the background as soon as you open the program. You can access the Vistory by clicking on the V button in the toolbar here, and you'll see it open up. And what the Vistory is it is a time-ordered series of automatically recorded events that you can then save and use as a record for what was done in your project. So you don't need to do anything to make this happen. They get populated automatically, and the general sort of rule for what goes in here is anything which has generated data, modified data, or done anything, any kind of filtering or statistical test should show up in the Vistory. So in here, you can see that from having loaded this project, I've got a whole raft of things in here where the data sets have been added. There was a quantitation done. I've created some replicate sets. I've done some statistical tests and created some filters. And I didn't need to do anything to make this happen. It just automatically came up when I started the project. Now, Vistories are intrinsically linked to the program. You only have one Vistory inside SeqMonk, and you can close and open this as many times as you like. So there's only one copy of it. It doesn't matter if you close the window. You can keep bringing it back. The idea with the Vistories is that they give you not only the set of things that happen to your data, but also enough context to be able to interpret anything that you did later on. So for example, if I look down here at some of the filters that I've done, I did a quantitation, and then I did a filter, which was, in this case, a DE-seq filter. Because the DE-seq filter used my quantitation that had happened at the point at which it ran, within there is not just a description of what DE-seq did, but also a description of what the quantitation was at the point that it was run. So that's the intention with this. Now, we can show the generation of a new one, new Vistory event on here. So if I go and make a new filter, let's just take my all probes list here, and I will say filter on values, my individual probes. So I'll just go and pick anything where the wild type had a value of more than 2. Okay, doesn't really matter what I'm picking, but if I do that, I generate my new list. And now if I go back and look in my Vistory, I'll be able to see this new event that was generated. So I filtered for places where exactly one of my wild types had a value above 2, and because that was a quantitation which used the quantitation, the filter that used the quantitation, then it records that as well. Yeah, Corinne, you've got something, question. Hi.
Hey, I made a comment as well.
Oh, yeah, sorry, I've lost the chat window somewhere. I'm going to try and find it. Okay, so sorry, please feel free to go ahead. Ah, there we go, yeah.
Yeah, so it seems like the Vistory is not showing up when you're opening it for us.
Oh, you can't see the Vistory at all. Ah,
No.
okay, so that's probably because Zoom's being too clever. Okay, give me a sec. Let me stop sharing that then. Let me do a slightly different thing. Oh, okay, let's not do that. It probably means, let me have a look what options I've got to share my screen. I might have to share. Okay, so if I just share the whole screen, that will do it. But unfortunately, you're going to see more stuff. So now if I do that, now does it show up?
Yes. Yeah,
Yes. Okay, cool. Okay, so because I now only have one screen, that means I can't see the things for the meeting controls. So let me just bring that back. There we go. Right. So can you just see SeqMonk and the Vistory up now?
yeah, looks
Yep.
good.
Brilliant. Okay, cool. Sorry about that. Okay, this is the first one. We're learning stuff as we go through. So yes, we've got the Vistory then. You can see the existing events that were in there that I didn't have to do anything to generate. And at the bottom, I have the new one that's there. Now, on its own, that's kind of useful, but it's also not as useful as it could be, in that it's just a bit of stream of consciousness at the moment. So things that will happen is that there may be things that you do and you decide, actually, yeah, I went down a blind alley there, or I don't want to go back. And also there's going to be times where, or certainly what you will want to do, is to annotate around this. Inside the Vistory, the only things that are recorded are automatic events that change your data. But anything else that you do that you might want to keep, so if you draw interesting figures and graphs or make tables of data, those wouldn't show up. So what we're going to look at is how we can annotate around what we've got already, and how we can insert other information and make this into a proper report. So if I look at what I've got now, the most obvious thing that we can put around here is commentary. So on here, at the top of the Vistory, I've got buttons to insert either titles, subtitles, or text. So at any point in the document, I can put that in. So if I click anywhere in here, you'll see I can select the individual blocks. And if I insert something, it will insert underneath whichever block I've currently got selected. So if I select the top one and say insert a title, okay, so I can say Vistory example. I can move blocks up and down. So on the right-hand side, you'll see that these little blue arrows that allow things to move up or down. I can only move things up or down that aren't automatically generated events. Because all of the rest of these have to occur in a particular order, then it doesn't make any sense to be able to move them up or down. I can delete things. So if I've done a bunch of stuff that I don't want to record in the end, I decided it wasn't worth it, I can press the red X's to remove it. But only things that I've inserted myself, I'm allowed to move up or down inside here. I can then put text in. So here I can put any old bit of commentary I want. Okay, and in a real analysis, you would write something generally before and after any particular sort of step that you'd taken to say what you were intending to do, then to do the actual analysis, and then to record what you think you saw when you looked at it. We can put as many levels of text and titles into the document. And it's important to do this not just because it sort of enters things into here, but you'll also see that when you generate the report at the end, that the titles and the subtitles are clickable sections in a table of contents, so you can quickly leap to any point in the analysis that you have tagged as either a title or a subtitle. Before we go any further, it's probably worth talking about saving and loading Vistories as well. Oh, hello, Joe. Hello, Joe.
Hello. Just to say that Stephen's trying to get in, and it says he's waiting for the host to let him in.
Oh, okay.
I don't know if you've
There
got
we go.
the ability.
Oh, I might have had some others, yeah.
Oh, and lots of others.
Yeah, okay. Sorry, I didn't realize they stacked up in the waiting room once we got there. Okay, so welcome to anybody who has been stuck in the waiting room. Sorry, we didn't see you there. Yeah. You haven't missed too much. So we've given you all we've done so far is the basics of getting started with Vistories and explaining that they are an automated way to record events inside SeqMonk. We've looked down the list of automated events that were generated in this project that have been done so far, and we're just looking at adding titles and pieces of text to them so that we can start annotating what we've got. We're just going to look at saving Vistories. So Vistories don't save with your SeqMonk project. They are separately saved documents that have their own format. They're not intrinsically linked to one SeqMonk project. So in theory, you could have a Vistory that you do part of the work in one project and then close it, open it up again in another project and annotate from there. But that's relatively unusual. It's much more usual that you might have done 2 or 3 different analyses on the same SeqMonk file and record those in different Vistories. So if you're looking at different aspects of the data. So to save your Vistory, you just press save. In this case, I've saved it before, but save as would allow me to give it a different name. It saves under these SMV files in here, and they go separately to the project itself. If you want to load a previously saved Vistory, you can just press the load button. Select it. When you do that, you'll actually get the choice as to whether you want to append the events in the saved Vistory onto what you've done already, or if you want to wipe out everything that you've got and replace it with the contents of it. So depending on which way you want to go, both of those might work nicely. If you have done some work in a Vistory and you haven't saved it when you exit the SeqMonk project, then as long as you've actually looked at the Vistory in that session, you will actually get prompted by SeqMonk to say, oh, you haven't saved your Vistory, do you want to do that? But if you've not interacted with the Vistory at all during that SeqMonk session, then even if it's recorded things, then you won't actually get prompted for that. So it only bothers you with prompts if you've done anything with the Vistory in that session. Let's have a look at putting some other features into the Vistories. So obviously the automatic features will end up in there anyway, but we can put other things in. So the main other things that we're going to put in are going to be figures and graphs. You don't have to keep the Vistory visible when you're doing this, so it's quite nice just to get rid of it, draw the graph, and then you can insert it and reopen it, and it will be there when we get there. So I'm going to take the example of doing a scatter plot. So if I go into plots and scatter plot, and I will draw a nice little scatter plot of these 2 data sets, and I decide, yep, that's an interesting thing, so I want to put that into my Vistory. If I go to save image on here, I'll actually get a choice of 3 places that I can save it. I can copy it straight to my clipboard, I can save it out to a file, or I can embed it directly into my Vistory. So if I just press Vistory on there, and then I open my Vistory again, you'll see now that this graph has been saved in here. If you had selected something in the Vistory, the graph will be inserted immediately below it. So in this case, it's actually ended up at the top of the Vistory, which maybe isn't what I wanted, but if I didn't want that, I can just use the X to get rid of it, click on the bottom of the Vistory, and then just save it again. And now it will embed into the bottom of my Vistory. If your graphs are or your figures are too big to display in the Vistory, so now I've just squished this up so you can't see it, you can click on the graph and slide left and right, and it will scroll across so you can see the remainder of it. So you can always see everything in here as you put it in. Okay, so that will be to insert a figure, and every figure panel that you bring up inside SeqMonk will always have an option to save, and that save will always give you an option to put it into the Vistory. The only place where that's slightly different is if you want to put in an image from one of the main views in SeqMonk, so either the genome view or the chromosome view. You can do those as well. If you want to put those into the Vistory, then you need to go through the File menu. So if you say File and Export Current View, you can select either the chromosome view or the genome view. So if I were to put the genome view into the Vistory now, that will just insert underneath, and I can see the genome view going in. Now, there's a couple of other little tricks that are useful to know about this. So one thing that's appeared quite recently in SeqMonk is the ability to annotate probes in some of the plots when you put them into a Vistory. So if I do a plot and go to a scatter plot, when you have plots like this, often there are individual points that you might want to highlight. Now, you can already do things like highlighting lists, so I could put on my list of interesting genes, and that's fine, and that would be good, and you could do that. But sometimes it's worth actually annotating individual points. So what you can now do is that you've always been able to put your mouse over a probe to be able to see what it is, but now if you click on it, that label will persist. So I can start labeling up interesting points in here, and those labels will stick. If I do that, if I want to get rid of them, I can get rid of them individually just by clicking on them again. Or if I want to get rid of everything, I can triple click. So double clicking on a point will show me the data in the chromosome view, but triple clicking will remove all of the labels. So I can use this to annotate up interesting points in here so that if I want to then comment on them in the analysis, those that are there. So now if I save the image and put it into the Vistory, I've actually got those points written on it, okay, which can be really useful. There's a related kind of trick in the chromosome view as well. So here, if I'm looking at this section of chromosome, maybe there's some genes here that I'm interested in, and I want to be able to show those when I'm exporting them out. I've got a couple of options. One thing I can do is to just put all of my labels on. So under the view menu, there is an option to show all labels, which you can either turn on there or say Control L, and that will turn all the labels on here, okay, which can be a bit of a mess. It never draws labels over the top of each other. So here I might want to label this gene, but I can't actually see it because this gene got labeled first and it won't draw the labels over the top. So it does show me a lot of stuff, but often it doesn't show exactly what it is that you would want to see. So if I turn those off, if I wanted to be more specific and I just wanted to say, well, look, this is the gene I was particularly interested in, so this is the one I want to show, I can make it highlight by just putting my mouse over it. But a little trick you can do is that if you use the Control X shortcut to create the chromosome view image from this, it will create that image with the highlight still on. So now if I save it to the Vistory, although it immediately appears to disappear there, inside the actual Vistory, that gene will still be highlighted. Okay, so you can put anything in, and with a couple of little tricks, you can also sort of highlight and label things as they go in. Other things that we can do inside here, we can also put tables in. So anything that brings up a table of information. So here, if I go to reports, let's say I do a data store summary report, just my basic one. Any table that comes up like this will have an option to save to Vistory. So if I do that, again, in my Vistory, that table will now be inserted. If it's a really big table, I'll have a scroll panel that goes inside here so that it doesn't just completely take over. But this small one will go in directly. often what we're doing when we put these tables in is that we are trying to show the results of statistical analyses and combined statistical analyses. So another thing that's useful to know about this is just to have a look at some of the options for annotating up tables in here. So again, this is something that's changed relatively recently in SeqMonk, and where we have more flexibility than we used to. So as an example, what I've done in this project is I've done a fairly standard sort of RNA-seq analysis where I've done a DE-seq differential expression to show that I got sort of 2,266 genes that change between my two conditions. I've got an intensity difference test, which is magnitude of effect test, effectively a sort of normalized fold change to find things that change a lot. Of which there's 274, and then I've got the intersection of those two lists, of which there are 256. Now, inside the DE-seq list, there are some interesting things that I want. So there is the p-value from the DE-seq, the false discovery rate, the log2 fold change, and the shrunken log2 fold change. Likewise, in the intensity difference, there is the p-value, the false discovery rate, and the z-score difference, okay, all of which are useful. But the list that I want to export is the intersection of the two, where I've combined these two together. And that list doesn't have any annotations on it. So if I just make a report out of this list, okay, and do the normal things, so I'll find the name match gene for each probe, I can see all of the expression values, I can see the probe names, which will be the gene names, and the details of the genes, but I haven't got any of the statistical measures in here. So if I'm going to report this, whether I'm going to put it into a Vistory or put it straight into a file, it can be useful to transfer those annotations across. To be able to do that, if I go into Reports and Annotated Probe Report, I'll still do the same name match gene as I did before, but now I'm going to look at this bit at the bottom that says Select List Annotations. So by default, it's going to put on any additional values that were recorded on the list that I'm trying to report on. So if I'd done the DE-seq one, it will automatically add the DE-seq parameters to it. But I'm not, I'm doing it on a list that doesn't have anything. So now if I click on this, I can see all of my probe lists here. So from the DE-seq one, I can see I've got the p-value, the false discovery rate, the log fold change, and the shrunken log fold change. So if I select all of those and say add those annotations, they will go in. And if I do the same thing on the intensity difference, I can add the annotations from those as well. So now I'm going to get all of the annotations from both of these lists, but reported just on the combined list. So if I do that, now I can see I'm getting 7 annotations on this, as well as the usual stuff that I would get. So now I'm getting my list of, 256 different genes, but within here, I've got the DE-seq hits here, and I've got the intensity difference ones here, and all of those will end up on here. So if I sort these by the DE-seq and then I save this into the Vistory, then in my report on here, I'll see now it's got too big to fit into the table, so it's got a little individual scroll bar that it can produce. But all of those annotations have gone in here. Okay, so it's a nice way of being able to annotate that up and putting it in here. Okay, let me just save this again. So I'm going to save this before I do anything else. One other thing that can be really useful to know about in these kinds of reports is that when you first set up a project, when you've loaded a load of data in and you've made up your replicate sets and you've changed all the names and just done all of that housekeeping that you do at the start of your Vistory can end up a right mess, because you end up generating lots of little individual events, one event for every data set you import, one event for every time you change the name on something, one event for every time you set up a different replicate set. Okay, and that can get to be a lot of things. So to make your life easier for that, what I often do is that instead of taking all of those individual events, is that we have another option in the Vistory here where we can insert a summary of the current state of the project. Okay, so if I look up on here, there's a button that says summary at the top. And having done all of that initial process, and I'm doing it at the end of this one, but normally you do it at the start, I can just press the summary button and it will insert a bunch of automatic events in here. So it gives me a thing with the basic project information. So what's the SeqMonk project, what Vistory project, what genome, what SeqMonk version, a list of all of the data sets along with their import options, a list of all of the replicate sets that I've made up. So I've got CKO and wild type and the names that go in there, and the current quantitation and all of the current filters with all of the descriptions in. You may not need that depending on what you've done so far, but it's a much cleaner way to start a project than seeing all of the individual events from the individual imports. So normally what I would do is to import all of my data, clean it up, get all the names and the replicate sets right, then clear my entire Vistory and insert a summary, and that's a good place to start this from. There's one last thing we can do with the contents of our Vistory, and that is that in addition to being able to put sort of text and headers and things from SeqMonk, we can also insert arbitrary content from outside the program. And this has actually ended up being really useful as well. So some things that are really obvious that you can do with this, if I had set up a particular filter, we'll get a description of what happened automatically generated by the program, but sometimes it's really useful to see what the dialog box itself looked like when I did that. So if I'd gone to this and I'd set up my quantitation pipeline and I decided, yes, this is how I'm going to set up my RNA-seq pipeline, I will get a text description of that, but I could just use the print screen. So if I do Alt and print screen, this is just a Windows thing, it will capture the current window. And now if I go back to my Vistory, I can just say paste image and it will insert that dialog into the Vistory. So I can see exactly how it was that I had it. And I can do this for anything else. So if I, for example, find a really interesting gene, so if I have my scatter plot up and I decide, oh, sorry, let me go back to everything. If I have my scatter plot up and I decide that, yeah, this gene here, this MOBP, that's a really interesting gene, and I'm likely to want to do something with that, I could go and search for this, go and find something like gene cards, to go and find the information about it, say, okay, what do I know about this? What's interesting in here? And I can just take a capture of anything that I want. So in this case, if I just take the snipping tool out of Windows, I can say, yeah, that stuff looks really interesting. Take a capture of that. And again, if I now go back to SeqMonk, in my Vistory, I can just say paste image, and all of that will go in as well. So I quite often use this for doing things like gene ontology results, that if I've done a gene ontology search, if I've gone to somewhere like gProfiler, just to capture the top of that, paste that into the report. It's not a complete record of it, but it's a nice little addition to be able to give some context around what you've got, or taking snippets of papers and inserting those. And it just gives you a bit more flexibility with what you can put in. Okay, so the last thing then, I guess, that we want to cover in terms of the functionality of the Vistory is actually rendering this into a report. So, I mean, your Vistory itself is a standalone savable file. You save it to an SMV file, and you can load that back in. I can just press the load button in this project or any other project and restore it from there. But normally what I'm going to end up doing is saving this as an HTML file. So if I click on Save HTML, it'll ask me where it wants to go. I'm just going to save it into my desktop. And it will then save that file and then should automatically open my web browser and will give me a rendered web report of the Vistory. So this is just going to be a nicely formatted single file that I can now give to someone else, save into a lab book, email to someone else. We've used them for our help desk system where we would upload them to there. All of your titles will now end up as clickable items in this table of contents here, where you can click through and jump to the appropriate place in the Vistory. All of your tables will have been inserted in here. If the table again is too big, then there'll be scroll bars on it, so you can scroll either left, right, or up and down in it, but all of the data is embedded in there. If you had images in, again, you'll be able to see those. If they're too big, you'll be able to scroll across. But all of this data for both the tables and the images is directly embedded into the HTML. So it's a single file that you can then share. Okay, so at the end of the day, this is what you would use to keep as your sort of permanent record of what you did in your analysis. The final thing I want to show you then is something that we've built on top of this. Because these kind of Vistories are a really easy way to demonstrate in a very quick way how to do something in SeqMonk, we always want to sort of put more examples out for how to analyze particular types of data. We've got some videos up on our YouTube channel, but videos are quite sort of laborious to do, and yeah, they need redoing every now and again. So a new thing that we've put together is on our website. So if you go to the bioinformatics homepage and then go to Vistory DB, this is a system which is a collection of Vistories that we've used to illustrate different types of analysis or different problems within SeqMonk. There's not a huge amount of stuff here at the moment. We've put some things in, but there's more that are going to come. So if you have any requests for things that you would like us to put into a Vistory and insert into the database, we are very happy to take requests to see what we can do with them. But on here, you can either just sort of search on here. So you can go and find things relating to RNA. So I think for sex-determinant RNA-seq or pairwise RNA-seq differential expression, or you can click on any of the predetermined filters in here, or you can just scroll down and have a look. But for each of these, if you click on this, there is a Vistory then behind there, which is exactly what I've just been showing you. So this is a slightly more complete one, doing a two-condition RNA-seq differential expression. All of the same sort of things as we saw before, so you can click down to look at the different sections. Here I've put in a lot more commentary, the images supporting what I've done, and the text at the end to say what I think it shows. And this is how we're now doing most of our analyses and how we're communicating them back with the people that we're working with. But they're a really sort of natural way then to be able to send the data back. Okay, I think I'm going to stop there because we've gone sort of 40 minutes. Hopefully you've got a good feel for what these are about. There's more information in the SeqMonk help about Vistories. There's a video on our YouTube channel which also goes through many of the same things that I've shown you here. And as I said, we will aim to put the video of this session up and make that available. We'll put it up on the same web page that the rest of the Bitesize Bioinformatics have been going to, and that should be up hopefully within a few hours of this finishing. If anyone's got any questions now, I'll leave both the chat and the call open, so you can stick your hand up if you've got questions or type them into the chat. But if not, then thank you very much for coming along, and we'll hopefully see you at one of the future Bitesize Bioinformatics. The next one is 3 p.m. on Thursday, when Felix is going to be talking about Nextflow pipelines and how we've been developing those at Babraham, and how they are now going to become a staple part of what we're doing in our high throughput processing. Okay, so I shall open it if there's questions. So yeah, either stick your hand up or stick stuff into the chat if you have questions. Hello, Corinne. Yeah. Oh,
Hey,
yeah. Hello.
I was wondering, so you can save all the plots that you make and all the data tables basically into your Vistory, but
Yes.
you'll still also save those separately to be able to import them, for example, in other analysis programs, or to edit the figures, or can you export that from the Vistory as well?
You can save them out of the Vistory. At the moment, the figures that go into the Vistory are all just PNG format, so they're fine if you're just going to want to stick them into like a presentation or something, but they're not going to be suitable for putting in as sort of publication quality figures. One of the options that we're almost certainly going to add in the near future is the ability to embed them into the Vistory as SVG files instead of PNG. Which is what they're doing at the moment. And at that point, you could then extract them out from the Vistory in a format which would be suitable to adapt for publication as well. So yeah, in the thing that I showed you, let me. Oh, actually, I need to share this again. If I just share that. Share. Oh no, sorry, wrong thing. I want to. So yeah, like in here, if you have a plot, you can just right mouse click and then save image as, and that plot will come out. They'll all get called index.png by default, so you just give it a different name. So, and that will then export that out into an individual file.
And for the tables, in what format could you export
So
them?
tables should copy and paste out if you want to do that. So because they're proper HTML tables in here, if you just select and copy, and if I take Excel and I just paste into that, if I do that, it should just copy and paste straight into there as well. Okay,
And
so
for the really large ones where you have to scroll through.
yeah, so that should work as well. So if you select the top of it and then scroll to the bottom, oops, that was a bit too far. Scroll to the bottom of here. Where's it gone? Yeah. And do shift and click on the bottom of it. That should select the whole lot, and then that should paste into there. Yeah, there you go. So that will also go out, so you can get that from there as well.
Okay, nice. Thanks.
Yeah. Okay. Right. Okay. If there are no more questions, then we'll finish the session here. Thanks for coming along. If you've got any questions afterwards, you can just contact us via the Babraham Bioinformatics website. All of our contact details are up on there. We're very happy to answer questions about this afterwards, but if not, thanks for coming along, and hopefully we'll see you at another one of these bite-sized bioinformatics sessions.